So welcome everyone back to the Genome Application Symposium. Now we'll have a session on genomics adaptation, challenges and approaches that will be chaired by Maria José Ruiz Lopez and Camille Cornet. So thank you both and looking forward for the discussions.
Mari, you're still muted.
Yes, you're still muted, Mari.
Good morning, everyone, and welcome to our next session, Genomics of Adaptation. I'll be presenting it with Camille. And we ended up our last session talking about differences between no-track genetic diversity and adaptive genetic diversity. So that is the perfect setup for our next session. So what we wanted to do, if I'm able to share my screen, which I don't know why it is not allowing me to share my screen. Give me a second.
Should be able, let me check.
It is not allowing me. I don't know why.
Luisa, can you check that? Because I'm.
Yeah, Mari is co-host, and you should be able. Yes.
Yeah, but I'm like, it is just like give me like warning messages all the time. And like, wait. No, it's not allowing me at all. I don't know why. Oh, now, finally.
Does it work?
Okay, now I think you can see my screen, right?
Yes, yes.
Yes, finally. Sorry for that. So, genomics of adaptation is a very broad topic, and adaptation is one of the key questions in evolution, and it also has ties to conservation of biodiversity and bioeconomy. So we wanted, Camille and I were thinking a lot about how to approach genomics of adaptation, because this is such a broad topic, which has such a breadth of like interest, and then also tools that can be used. So we wanted to make this session interactive. So what we are going to do, we are going to be using Mentimeter as well to gather some of your insights on the challenges and approaches that you are using. And then we are going to highlight two of the case studies that we are doing within BGE. And hopefully we can get a conversation going on, mainly about how high quality reference genomes can help us to gather more information and overcome some of the challenges, but how that brings some challenges as well, for example, with quality of annotation, etc. So we will be dividing the session in two areas. First I'll talk, then Camille will talk, and we will interact with you through the Mentimeter. So the first thing, if you have here the link, maybe you can copy it, and we have questions like, when you think about genomics of adaptation, sorry, that's not, why it is not coming back? When you think about genomics of adaptation, what's the first thing that comes to mind? Maybe if you have the link, you can start answering that question. Adaptation to what? What is what you think? Because this is such a broad topic, and we wanted to know what were your thoughts. So we have selection, genetics, local.
I just want to say that I don't know if it's on purpose, but we don't see the word cloud popping up. We just see the QR code still.
You are not seeing I have it in presenter mode. Let me,
Maybe you need to share another
Yeah,
screen.
again. Yeah, I'm having a lot of issues with that person with the sharing. Okay, give me. Are you seeing it now?
Yes, perfect. And there's the code there as well that I will copy in the chat for the others.
Okay, so I'll give you a couple more minutes. And we have 47 responses already, so we are reaching, I think we are reaching the limit. 54, great. Yes, I think this cloud that is popping up, it really shows how broad the topic is. Because the first thing that we are thinking about when we do the genomics, it goes from the tools that we are using and how can we actually measure selection and study selection and study the genomic footprint of adaptation to what kind of adaptation we are looking at, from local adaptation to environment, climate change, anthropogenic human footprint, anthropogenic pressures, etc. So this really shows the breadth and how interesting this topic is and how broad it is. So I think, yeah, I'm going to give you 1 more minute so we can keep this going. So we have stressors, mutation, gene expression, etc. Structure variation, genomes, environment, temperature, which is very local conditions, local habitat. Selection and environment are the ones that pop up more, but we have like this really nice cloud with a lot of little things. So thank you very much. And now I'll try to share the other screen. To go back to the presentation. Yeah, so as we saw, it is really, really broad. Great, Camille is telling me. So we saw it is really, really broad. So what we are going to do now that we know like what are the main interests of people, like we are going to highlight 2 case studies of the VGE project that look at adaptation in different ways and are like different topics of adaptation and use different approaches. So I'll start with my case study that is analyzing the genomics basis of vectorial capacity and insecticide resistance in Culex species using reference genome and population genetics. So vector-borne diseases are one of the major concerns in public health because they are increasing, and they are increasing due to different factors, including environmental factors and climate change. One of these diseases that is increasing its incidence is West Nile virus. So West Nile virus, it's an emergent disease here in Europe, and it's the most widespread flavivirus around, one of the most widespread flaviviruses around the world. So it is transmitted by mosquitoes to birds in a zootic cycle, and it occasionally affects humans and horses who can develop a disease. One of the major problems to study West Nile virus is that it has a very complex ecoepidemiology, where we have a lot of factors influencing the virus transmission, including which are the vectors, and the reservoirs. And this is going to influence when it spills over to the accidental host, that are the humans and the horses. But one of the things that happens is that West Nile virus can be transmitted by many vectors that belong to the species Culex. For example, in our area, that is a hotspot for West Nile virus and has had 2 outbreaks, one in 2020 and then an ongoing outbreak in 2024, we have 5 different Culex species in the area, but we don't know exactly what makes them good vectors of West Nile virus and how are they adapted to the environment and how they are adapted to the virus. What we know, though, is that 2 of those species are the main drivers of the West Nile virus transmission. One is called Culex exiguus, which inhabits mainly natural areas, and it's the one that amplifies the enzootic cycle inverts. And then we have Culex pipiens, which is the house mosquito, the common mosquito, which helps in the spillover to humans in urban areas. So as you can see, there are clear differences in how these different species of mosquitoes interact with the virus. So our question here was, okay, so we want to, what are these differences? We can study this with vectorial capacity, which is a very complex formula, which depends on different factors: vector density, vector biting rate, pathogen-restricted incubation period, and probability of vector daily survival. And this determines how capable are vectors to transmit a disease. But it also depends on vector competence, which is mainly a genetic factor. So we thought that genomics could help us understand a little bit more what is the different vectorial capacity of these 5 different species. And how can genomic tools help us? And how is this relative to adaptation? So on top of dispersion rates and population structure, genomics can help us to understand patterns of local adaptation, whether some populations are more adapted to certain temporal characteristics. So for example, in Spain, there is a clear gradient of temperature from south to north. These species inhabit the whole peninsula, but we only have West Nile virus in the south. What is the vector competence and immune? For example, what is the immune response of these vectors to the different pathogens? What is the host preference? These 5 vectors that I was talking to you about fit on different animals, and this is going to determine whether they are able to bite a bird that is infected or not, and we can study that through the evolution of chemosensory or olfactory receptors. And also, it is very important because we are in an outbreak area and the pests, these insects need to be controlled. We are using a lot of insecticides, which is one of the major examples of rapid evolution of rapid adaptation. So the evolution of insecticide resistance can also be studied using genomic tools. So one of the first studies we did looking at how a vector can adapt differently to different pathogen species was looking at Culex pipiens and how it is able to transmit and respond to the different Plasmodium species. And we did this in Culex pipiens. And we used transcriptomic tools, which is one of the tools we can use to study the genomic basis of adaptation. And what we found was that this species was responding different to the 2 avian Plasmodium species. But what I wanted to highlight here is that was one of our major challenges. And it was that we didn't have a good high quality reference genome for Culex pipiens. And we had to map it to the related Culex fasciatus. It wasn't well annotated. So we know that even though we got some of the information, we lost part of the information because we had genes that were expressed that we didn't have the annotation for. So that is one of the main challenges that we are finding, and I wanted to highlight that because we will talk in later about challenges. Like in our case, it was the limited genomic resources for Culex mosquitoes, with only one high-quality reference genome available for Culex fasciatus. We can see here in the graph how other species like Anopheles have much more genomic information available, which makes genomic studies easier. So the goal of this BGE study was to sequence high-quality reference genome for the 4 Culex species that I mentioned before, and the Culex pipiens that became available through the Darwin Tree of Life a couple of months ago. And then the first aim is to use comparative genomics to understand vectorial capacity and vector competence. So here, wanted to highlight the different approaches. So what can we use to study adaptation? So on one hand, we can study at the species level, we can use comparative genomics, but we can also use population genomics to study other factors. And in this case, we are using as well whole genome resequencing and population genomics to understand the evolution of insecticide resistance across times for Culex pipiens and Culex perexigos, where we are going to be looking at signatures of selection and selective sweeps, specifically and candidate genes for insecticide resistance. So this is a little summary of the project that kind of like encompasses like two very different approaches to kind of a similar question. So I'll pass now. Yeah.
Thank you so much, Mari. It was super nice. If anyone maybe has a question relating to Mari's topic before I start, we can take that now. Otherwise, I will share my own screens so we can continue. Right. Can anyone tell me if we can see this well? All right, thank you very much. So, well, it's super nice, I think, that we got to co-chair this session together, Mari and I, because we have like projects that use similar approaches to understand genomics of adaptation, but with like different goals and systems in mind. And I think these like kind of merge well together. And as Mari said, it's also super nice that we got this session just after the fantastic session of Camilla and Ivana, because, well, it finished on a note about how using adaptive variation for conservation genomics purposes is still light years away. And maybe that's true, but I hope that together today we can maybe do like one step forward at least towards this goal. And I want to start with another example or a BGE case study that we got. Thank you very much to BGE, by the way. So we were interested in conservation of alpine butterflies in the face of environmental change, so mainly climate change and land use changes. So imagine you have butterfly species that has this suitable green habitat, and well, I'll highlight why alpine butterflies are particularly sensitive to environmental change, because you can imagine that if temperature rise and habitat loss happen, in one scenario, this would lead to smaller and more isolated population as the suitable habitat range goes up in elevation and becomes more patchy and separated by the valleys in between the mountains. So this would lead to loss of genetic diversity and connectivity between population, and potentially species would become endangered. But you can also imagine the scenario where where the populations or the species might be able to adapt to those changes and cope with them. And I think it's super important to be able to assess if our species of interest is more likely to undergo the upper scenario or the lower scenario, to be able to make informed conservation priorities. And I strongly believe that genetics can help us with that, and this is what I'm going to try and highlight now. The genus we're focusing on is Erebia, and I think it's a perfect system to explore those issues, because they're often alpine species that are cold adapted. So that makes them particularly sensitive to climate change, and they have a hotspot of biodiversity in the Alps, where a lot of secondary contact zone and cryptic species coexist. But we also have the possibility to compare some certain species that are very common and widespread all over Europe, while others are already very rare and restricted, like the beautiful Arabia Pluto you see on the picture on the right here, which is occurring only like above 2,000 meters and is already encompassing several very isolated population, and it's potentially a cryptic species complex. And I think that's a very important issue that Ivana touched upon, that of course, if conservation practices merge together in a species level, a lot of different subspecies or population that are very distinct, well, it might give us a wrong idea of how endangered they are. So now how we want to assess adaptive potential, well, I don't think I need to convince anyone here that having a good quality reference genome is very important. And so that's what we started doing by generating reference genome for 12 Arabia species. And here I have to acknowledge Project Psyche that we're involved in, that's aiming to generate reference genome for all the Lepidoptera in Europe over the coming years. And so thanks to them, we can have like a lot more Arabia species to explore. And on top of that, we have or will have population genomics data for both widespread and restricted species to be able to compare. And this type of data can already give a super important variable to, like we discussed just before, like that are important for conservation genomics, like genetic diversity, connectivity between population, and like resolving cryptic species complexes. But we would really like to go further and to assess the adaptive diversity that might be important for these populations and species to cope with climate change. And for that, here I want to insist on not only having good quality reference genome, but also having good quality reference annotations to be able to understand where in the genome is the important variation and if it is neutral or not. And this is where we got this BGE case study to do RNA sequencing to have those reference annotations. I also want to mention that having good quality genomic data is very good, but it's not sufficient for conservation. And here I want to highlight Infofona, the Swiss National Center for Phonistic Information that we are working closely with, that are doing long-term population monitoring in the field to be able to assess exactly how the populations are doing at the moment. And also to be able to understand adaptive potential to climate change, it's important to know actually which genes are involved in cold and high elevation adaptations in Arabia. And that's a project that our friend and colleague Irena, who I think is here in the audience today, is really interested in. And hopefully we can join forces and look at this together. So this should hopefully help us to have an idea on the functional genetic diversity of the species and their potential to cope with environmental changes. Obviously, it's not that easy, otherwise we would not need an entire session on that today. And I hope that we can discuss to have better idea on how to do that later. And so here I prepared like a little roadmap on how we can go from our species of interest to a conclusion on their possibility for adaptation, for example, to climate change, but also it could be to insecticide, like Marie mentioned, or any other adaptation of interest. So first of all, we could make a good reference genome and annotation, generate some population genetics data, and do stuff like GWAS and things like that. Oh, something was drawn on the screen, no? Do you also see that? Anyway. And then from there, we could find the genes involved in adaptation that are potentially under selection and understand the functional diversity and adaptive potential of our species of interest and make a conclusion on their potential for adaptation. But of course, as I mentioned, there's still many, many challenges. It's of course not that simple. For example, at each level of this roadmap, there are some challenges like generating reference genome is relatively easy now for most species, but I think that having a functional annotation for non-model species is really not easy. And like the type of variation that might be important for adaptation is also not super clear. Like there's a lot of non-coding variations, structural variations, splice variants that are still difficult to map and to understand their phenotypic effects. And talking about phenotypic effects, of course, to be able to understand how diversity is actually functional and adaptive, we need to be able to make a link with the phenotype and with the fitness of the species we're looking at. And for species that you cannot keep in the lab and do plenty of experiments on is really not easy. And I think some areas that can help us maybe is other omics, like Mari talked about using transcriptomics. And something that's very important for me is the ecological aspect, because as I mentioned, having good genomic data is great, but without the ecological context in which your species and population evolve, it's hard to really understand what's going on, I believe. And so I left this roadmap very broad and even unprecise, maybe voluntarily, so that we can leave as much space as possible for discussion. And I would like to go back to Mentimeter to have your opinions. And unhelpfully, it's not the same code, not the same QR code as the one that Mari presented, but so I will leave you like a few moments to join. Is everyone managing to join the Mentimeter? Like this first one is just a random one. The real question comes later, but so that the QR code was not like on top of the answers of the next question. Okay, let's go. So the first question I have for you is, what approaches are you actually using in your adaptation genomics research on your species of interest? Comparative genomics, population, you can answer several of these, right? Population genomics, other omics, or something completely different that doesn't fall into those broader categories. Right, a huge majority of population genomics for the moment. That's kind of what I expected.
Camille?
Yeah?
Can I say something?
Yeah, of course.
While we wait. Yeah, because I mean, I found it interesting that you added comparative genomics to the Mentimeter, but you didn't add to your roadmap. And I think that could be something very interesting, in between the reference genome and the population genomics, the comparative genomics across genomes would be also quite important.
Absolutely, absolutely. I fully agree with you, and that's what we're trying to do as well with like the comparison between the widespread and more restricted species. And true, I should have found a way to fit it in the roadmap. Fully agree with you. I'm very curious.
yeah.
Anyone? Yes, Mari?
No, I was going to say that what Camila brought up, it's really interesting, because one of the things that we were thinking, like, how are we going to focus this? It was really difficult because there are so many different approaches that, so yeah, it was kind of like, I think it's really interesting to see like the perspective that everyone has, and we wanted to actually see that.
Absolutely. And I also, I want to ask maybe if someone that answered others would like to speak up and say what they mean by that. Very curious.
Or write it in the chat. It will be okay.
Absolutely, yeah. In the meantime, we can maybe go to the next and last questions. And this is an open question, because this is what really we wanted to focus on and to have an interactive discussion. What do you think are the biggest challenges in your research or in general to study adaptation genomics? We mentioned a few, like Marie talked about having enough genomic resources. I think personally, since I work on species that I cannot keep in the lab, it's linked to fitness. I would agree with that. Yeah. I believe a lot of people here are working on genomics of adaptation, so I'm curious. Yeah, of course. Sample collection for some species much harder than others, for sure. Yeah. Perfect quality samples, yeah. Eliminate confounding effects, yeah, for sure, like demography and so on. DNA quality, reaching a good sample size for GWAS, for sure. Cost and specialized equipment, technical support. A lot of people are mentioning sampling, and I think this is where, depending on your species of interest, might be very different. Like some people might be luckier than others, and yeah, for sure. Oh, didn't realize I could scroll down. Geographic scope. Yeah.
Small geographic regions with no so adaptive difference. Methods are heterogeneous.
Difficulty in measuring fitness and identifying relevant phenotypic environmental variable. Yeah, I very much agree with that. This is still like the area of the roadmap that I find super blurry. Like, how can you actually do that? Yeah.
So there are some of the answers that go along the lines of access to technology or data analysis capacity, which is very interesting and give us an idea of how complicated, like it's not only the question, but also like accessing the technology, financing.
Yeah, hopefully the like huge initiative and big sequencing consortia that are popping up at the moment can help with that by bringing together people from different geographical regions and means.
Oh,
yeah.
Some people is mentioning like lack of statistical power for identifying adaptive loci due to low sample size. And I think that might be one of the major challenges when you are studying adaptation in a small or endangered populations. You have smaller sample sizes, you have limited sometimes access to those samples. They might be inbred, which further complicates your studies. So that's also a very interesting point.
Absolutely.
Distinguishing plasticity versus adaptation.
That's a big question. I really like this one. Like the problem we face with human genome was not representing the whole population. Like, is the future then to have like pangenomes for your species of interest that might be representative of all of the population you want to assess? I think that might be an area that goes further. Accounting for the effect of demography, yeah, all the confounding factors, absolutely. So I'm really glad of all the answers that we got here. Like this is really interesting. I didn't expect to have so much emphasis on sampling, but for sure that's like the basis. And yeah, thank you so much all for participating. I think we can go out of the Mentimeter now. I can stop sharing so we can leave space for discussion. I completely lost track of the chat while I was sharing screen.
Yeah. So regarding, I'm going to go regarding the others, so going back to the Mentimeter, so what were others? So Rob Waterhouse added others, experimental evolution, obviously not applied, but more fundamental. So that is one of the others approaches. And then Gabors Ramko, he meant screening for outlier loci to identify putatively adaptive genomic regions. Probably it is within population genomics, but yeah, population genomics sometimes is very broad. So that would be two of the questions. So we have also questions for your talk and my talk. So I'll start with yours. Like
Go for it.
Alexander Nater, very interesting topic. What would you say is the best assembly strategy for such resequencing studies? A primary assembly without aware mapping tools, or rather a haplotype resolve assembly and then just randomly selecting one of the haplotypes for mapping?
Okay, that's a good question. I think that a primary and alternate assembly is still very much enough for these type of studies. But I would say depend maybe on how heterozygous your species is. I personally went for primary and alternate because it was sufficient, and also, well, when you don't have the possibility to have like parents and offspring to sequence to make a really well-phased reference genome and haplotype resolved, I don't see really the benefit of, yeah, I don't know, maybe someone has another opinion on that. I would love to hear it. Yes, Camila?
Camila?
And I can talk a little bit about our experience. So the haplotype results, in some cases, the quality has been amazing and the same for both haplotypes, in which case we do opt to use both of them in the analysis. But in other cases, it does happen that one of the haplotypes, and in our case was because of data, not the genomic complexity, and did not have the same quality. So yes, as Camille says, it's still, I think, an ongoing development process, and also for our understanding. But there is a lot of power in having two haplotypes, especially if they have differences. So we don't have trios or anything like this. We are simply using a HiFi data bundled with Hi-C data for directly phasing, or let's say pseudo phasing the haplotypes. And what I can say is HiFi data, when the coverage is high and the reads are longer, this has been working much better for us in this sense. But I don't think there is no answer, because it really depends on the genome. So I would go for a good representation of the genome at the haploid level. Yeah, so either the primary one or the best haplotype that you have.
Yeah. If you're working on a genome assembly project, it's definitely worth it to try both and assess if it makes I mean, if it makes a difference, or if you find that you have good quality with phased haplotypes, then definitely go for that. There is a question for you, Camila, actually. What do you mean by good coverage for HiFi data?
Yeah, I just answered. I think 40-fold or more. The last like 25 or 30, as we have been doing, it works well, but in comparison, a little bit more really makes a difference. But it might not be only about coverage, right? It could be about representation and read length and so on. But I can say that a little bit more coverage does help, but increases costs, of course.
So there was another question for my BE project that Marie Noel asked me about high quality reference genome and whole genome resequencing. Could you please tell us what is a high quality reference genome for Kulik? So I'm thinking about the contiguous accurate and annotated genome assembly. So the more like it can give us like the structural information as well. That's what we are generating. And with the whole genome resequencing, we are using a shotgun approach that when we are later mapping that to our high quality reference genome and then looking at the annotation. So that was the difference between the two approaches.
Thank you, Maria. I'm Marie-Noël. So if I understand properly, when you talk about a reference genome, it means there is no gap in the genome,
Yeah.
correct? Okay.
Yeah, and it's organized by the different chromosomes and you have all the information. Yeah.
Okay. Okay. Thank you very much. Is it the same with the butterfly? You are also talking about complete genome?
Yeah, so we have chromosome level reference genome, but we used Hi-C for scaffolding, so you still have some gaps in between the scaffolds. But we try to have a reference genome that as contiguous as possible.
Yeah.
Thank you very much.
But it's not telomere to telomere per se. It does have some gaps. Camilla, you want to add something?
Yeah, it's just because of the topic. So we try to reach as a minimum quality the requirements from EBP for what we call high quality chromosome level genomes. So there is a percentage of gaps that are still acceptable. Yeah, so it's not a complete lack of gaps, but it's a very low percentage that is acceptable for us to consider this high quality and already be satisfied for the comparative analysis.
Thank you.
José?
Yes, I don't know if Jacob will talk about
just to
this
comment on
issue
that.
of reference genomes, because I was going to move forward to population level data.
Okay, you shut up.
Can I start then?
Okay, fine.
Okay, then I start. So I think I was one of the person that responded sampling, because then, so we are talking about, and it's very important to have a high quality reference genome as contiguous as possible. But then for adaptation studies, well, if we are looking for adaptation, I don't know, to environmental gradients, then it means that we need to sample those environmental gradients. You mean to sample the species along those environmental gradients. The good news is that if we manage to do so, when we have then resequencing data, so not necessarily additional reference genomes, but short reads that we can map to the reference genome and get allele frequency data along the genome for these populations, we can find, for example, whole genome resequencing means instead of assembling de novo the genome of some individual, we produce short reads of which is much cheaper and faster. We produce short reads for a certain individual, a lot of them, which means that we cover the genome several times. But then we use the reference genome to map, to align all those short reads. In short, I mean 150, 300, or depending on the technologies. And we map back to the reference genome. And so that gives us allele frequency estimates along the genome for a given population. And so this has the potential to discover correlations of allele frequencies and environmental gradients. And if we do find that, we find loci which are actually important for the maintenance of adaptation to a certain environmental gradient, which if the environmental gradient is predicted to change, then certainly it will affect this adaptive status of the populations. We can also look at the phenotypes, right? And if we have a phenotype of large effect, as Mari is looking at the phenotype, which is with this vectorial capacity, then we can also get the genetic basis of that. And then I think that to mitigate a little bit the challenge that we were discussing, the trick would be to use this wealth of information of a genomic study to then design specific tests, specific, I don't know, SNP chips or easy assays that will actually monitor this type of variation that we infer from from the whole genome resequencing analysis. And so this would make it applicable and more amenable for practitioners. And I'll shut up. Jacob, do you want to pick up on this?
I added my comment in the chat, but I think this discussion on insisting on certain types of quality and certain standards is stupid. It depends on the research question and what you're actually after. So this discussion of whether or not we should go for EBP standards or whatever, it really depends what you're after and the limitations of the study.
I do think that
I just
EVP
want to add,
standards are...
sorry, the BGE,
No, go ahead, Camila.
project goes for EBP standards. That's the policy of the project. So
Go for it.
the usage then after that, the application for each one, then of course it depends on each project. And there might be many other projects where people might want to use something that is like a draft genome or a reduced representation. This does not prevent people from doing their analysis, but definitely our policy and our belief, let's say, is that a good quality reference genome will always make a difference in the analysis, especially when it comes to adaptation.
I agree, Camilla, but if you're insisting on EBP standards and always come back to that, you're putting off people that have less quality data from even trying to publish. And I think that's it's not a way forward.
I think this is another discussion that maybe we can have later.
We are also reaching the end of the session, I think. It's 11. So thank you very much. It was really interesting. It was really interesting to see all the different challenges that people is finding. What are your thoughts about adaptation and what are you using? It seems that population genomics is one of the major tools and all, well, everything within population genomics, because population genomics itself is a really broad. And it was really interesting to see all your topics. And I think it will give us a lot of very helpful information to keep us going and keep us like learning and moving forward on how to study the genomic basis of adaptation.
Yes, thank you so much for the organizers and for all of you for participating. Thanks. Looking forward to the rest.
Thank you, Mari and Camille. So we will break now again, and we'll come back at 11:15 with a session on one first dimension of the gap analysis that we are doing on the implementation of genomics in biodiversity applications. So thank you all for this discussion, and I'll see you back in 13 minutes. Okay, thank you.
Bye.
Bye.